Welcome to another lecture by MedicoMedics, Genetics Chapter 8, Cytogenetics. In this lecture, we will be discussing cytogenetics. What is it? We will talk about chromosome structure and function, karyotyping, mention some chromosomal abnormalities, some clinical examples. Continuing on, we will talk about some applications of cytogenetics and, as always, end with a summary. Now, cytogenetics is the branch of genetics that studies the structure and function of chromosomes. So focus areas are chromosomal abnormalities, techniques for analyzing chromosomes, and of course, clinical significance of genetic disorders. Now, chromosomes, structure and function. So what are they? They are structures that organize and store DNA. And humans have 20 pairs of these, 46 in total. What's its structure? Well, it has a centromere, which divides chromosomes into short and long arms. We have telomeres. This is the protective cap at the end of the chromosome. What is its function? It's to ensure proper DNA replication and segregation during cell division. Example, chromosome 1 is the largest human chromosome, carrying about 2,000 genes. So chromosomes are like bookshelves that store and organize DNA, so it's easy to find during cell division. Now, what is then karyotyping? Well, it is a visual representation of chromosomes arranged by size and shape. We use it to detect chromosomal abnormalities, for example, Down syndrome. It helps us identify gender, so is it XX or XY chromosomes? If you remember, XX is female, and XY is, you guessed it, male. And Down syndrome, for example, is also known as trisomy 21, where we have an extra copy of chromosome 21. So the process of karyotyping, we collect cells, so blood and amniotic fluid. We stimulate cell division, do a stain, and photograph the chromosomes. So, as mentioned in Down syndrome, karyotyping revealed that the chromosome in the extra chromosome in trisomy 21 in Down syndrome. So a karyotype, you can think of it as a photo of your chromosomes lined up to check for missing or extra pieces. Chromosomal abnormalities, different types. So we have numerical abnormalities. I think this one is aneuploidy, and if I'm pronouncing that wrong, live with it. So this means extra. You have an extra or missing chromosomes. So for example, in trisomy 21, you have an extra chromosome. So in Down syndrome, for example, and we call it trisomy 21. If you have monosomy, you have a missing chromosome, right? And this is an example of this is Turner syndrome, where you have 45 in total. What about structural abnormalities? Well, there could be deletion, so there is a loss of some chromosome segment, like in Cri-du-Chat syndrome. You can have duplication, where there is an extra copy of a chromosome segment. You can have translocation, where segment moves to a different chromosome. You can have inversion, segment flips within the same chromosome. So having these kind of chromosomal abnormalities are like books missing pages. So having extra pages or sections swapped with some other book, it's all mixed up. That's essentially what we're talking about here. Now, what are some clinical examples of chromosomal disorders? Well, we have numerical disorders like Down syndrome, trisomy 21, and we can have the clinical features of developmental delays, characteristic facial features, etc. We can have Turner syndrome, right, where there is a missing copy. Here we have short stature or infertility in females. Klinefelter syndrome, where you have an extra copy, 47 chromosomes. And this is produces male infertility and learning disabilities. Now, Klinefelter only applies to males, and Turner syndrome only applies to females. What are some structural disorders then? We talked about numerical. What is structural? Cri-du-chat syndrome, for example. And typically it's a high-pitched crying in infants, and you see developmental delays. Chronic myeloid leukemia caused by a translocation between chromosome 9 and 22, also called Philadelphia chromosome. So some of these disorders are caused by having too many or too few chromosomes, while others result from broken or rearranged pieces. So hopefully you have now an idea of how to differentiate between these disorders and these. It's actually on the level of the chromosomes itself. Here you have a issue with a quote-unquote broken or rearranged setup of your chromosome. Here you have chromosomes either missing or you have too many. Now, cytogenetic techniques. So we mentioned karyotyping, which helps us visualize entire chromosomes for abnormalities, like a photograph. We also have, the abbreviation is FISH, so fluorescence in situ hybridization. Here we use fluorescent probes to detect specific DNA sequences. For example, identifying chromosomal deletions in cancer. We have comparative genomic hybridization, detecting gains or losses of chromosomal regions. We have arrays, so higher resolution for detecting submicroscopic abnormalities. So, for example, fluorescence in situ, so FISH, is used to confirm the presence of the Philadelphia chromosome in leukemia. So cytogenetic techniques are advanced microscopes, essentially, that help scientists to see changes more clearly. Now, what are telomeres? So these are repeated DNA sequences at the end of chromosomes, and they help in protecting chromosomes from damage. Their significance: shortening of telomeres is associated with aging. Dysfunctional telomeres can lead to cancer. So think of telomeres essentially as a protective structure for your chromosomes. An example: cells in older individuals often have shorter telomeres, and this is linked to aging and cell death. So, yeah. Mosaicism or mosaicism, however you want to pronounce that, is the presence of two or more different genetic cell lines within an individual. So it's like having two types of cells in your body, each with a slightly different genetic makeup. Causes errors during early cell division in the embryo. Example is mosaic Down syndrome. So some cells have an extra chromosome 21 while others don't. Mosaic Turner syndrome. So some cells lack an X chromosome and others don't. So essentially it's a hybrid form of having trisomy 21 or Turner syndrome. You have both quote-unquote healthy chromosomes or cells, and you have the presence of extra copies or missing copies in other cells. What are some applications of cytogenetics? Well, in medicine, we use it to diagnose genetic disorders. For example, prenatal testing for Down syndrome. It helps us identifying cancer-related chromosomal changes. It's used in reproductive genetics, detecting chromosomal issues in embryos using IVF. In research, so understanding chromosome structure and function. For example, cytogenetics helps identify chromosomal abnormalities in embryos to improve IVF success rates. And IVF is in vitro fertilization. So cytogenetics is used in medicine to diagnose disorders, but also in fertility clinics to ensure healthy embryos, and in labs, of course, to study chromosomes. So in summary then, cytogenetics studies chromosomes to understand their role in genetics. Chromosomal abnormalities can be numerical or structural. Techniques like karyotyping and fluorescence in situ FISH are critical for diagnosing genetic disorders, and applications include medicine, reproductive genetics, and research. That's the end of Chapter 8. Continue now to Chapter 9.